the islet regeneration project, we work almost exclusively now with what we call mesenchymal stem cells. And we have developed techniques to deliver the mesenchymal stem cells directly to the pancreas. But cell therapies are a difficult thing to do, especially when you're trying to deliver cells to the pancreas. Most of the pancreas is an exocrine organ, not endocrine. Endocrine is the making of insulin, etc. So you don't want to disturb the exocrine portion of the pancreas, because that could induce a disease called pancreatitis, which can be fatal. And the reason being is that the exocrine cells within the pancreas, if they're damaged, they release a lot of digestive enzymes that could cause all kinds of trouble. This is why pancreatic cancer is so deadly. They can't go in and cut out the tumor in most cases, because because you can't surgically manipulate the pancreas very easily without causing pancreatitis. We figured out a way to deliver the cells directly into the pancreas. But what we're really high on now is, cells could be rejected. Stem cell exhaustion occurs. The cells in the sick patients may not be as functional as cells in healthy patients. And whenever you're doing a cell transfer or a cellular therapy, you have to worry about allogeneic matching so that the cells aren't rejected by the body's immune system. The patients need to be on immunosuppressive therapy. So there's a lot of hurdles to overcome for a cellular therapy. So what we did was we developed a way to stimulate a really important pathway in mesenchymal stem cells. We stimulated the Wnt pathway using a drug, basically, called a GSK3 inhibitor that blocks one of the major inhibitory pathways of the Wnt pathway. So really what we're doing is we're using a drug that turns on Wnt signaling in the mesenchymal stem cells as we're growing them in the lab. So we knew that what this mesenchymal stem cells were secreting, the proteins that they were secreting, were what was important for inducing islet regeneration. The mesenchymal stem cells do not, by themselves, change into insulin-producing beta cells. What they do is they secrete reparative and regenerative factors that can help the pancreas heal itself or help new islets to form. So we knew that what was really important was the proteins that were being secreted by the cells. And by stimulating the Wnt pathway, we were able to generate a cocktail that was more highly regenerative in our mouse models of diabetes. So if we took the regenerative cocktail, our Wnt stimulated MSC media, and injected concentrated conditioned media, which is basically the proteins from that the MSC are secreting as they're growing in the lab, we were able to induce almost the exact same thing that we induced when we injected the cells. So I call it stem cell therapy without the cells. We're now delivering a mixture of proteins. There's some microRNAs, there's some extracellular vesicles in there. We're interested in all of these different things. But really what we're trying to do right now is to use advanced proteomic techniques to figure out what proteins are in that mixture when we deliver them directly into the pancreas of the mouse that can induce islet neogenesis. And that's really where we're at now. We really need to find out what are the proteins that are important, what are the proteins that we don't need, and we need to find out how this regeneration is occurring. We need to know the mechanism of new islet formation that is induced by MSC. And we're working very hard in the lab at doing something called lineage tracing to find out what is the signal receiving cell in the pancreas that will respond to these proteins that leads to new islet formation. We've got a couple of specific mouse models to say, well, do the new beta cells come from alpha cells? There's been a lot of talk in the literature that alpha cell and beta cells have a plasticity. They can sort of go back and forth between the two phenotypes. And if you have something that will kill the beta cells off, the alpha cells will differentiate to replace those lost beta cells. So that's one of the candidate targets that we're looking at, to see if we can induce alpha to beta cell transition in these mice that have no beta cells, where the beta cells are destroyed by a drug. The other thing that we think is very important is that, well, if you're differentiating all your alpha cells into beta cells, what's gonna replace the alpha cells to keep this process going? And again, going to the literature over the past 10 years, it looks like processes like epithelial to mesenchymal transition are important. So we think that there are facultative islet precursor cells in the ductal epithelium, the ductal epithelium, that these ductal cells could be activated to differentiate into an endocrine-like cell, probably an alpha cell first and then a beta cell later. And that's, we're trying to use very advanced lineage tracing models to document that process.
Okay, that's actually
hard to explain.
That's actually really interesting. So just to summarize, so right now you're currently trying to figure out the proteins and I guess other extra factors that are currently in the media of the from the mesenchymal derived cells, and trying to figure out what I guess proteins are causing the transition back into beta cells. Correct?
That's right. What is pushing this whole process? It's sort of, I think of them a little bit separate because we have different people in the lab working on the different projects. We have a postdoctoral fellow who's a proteomics expert that's trying to figure out, well, what are the proteins that are important? When you do proteomics, you get like 3,000-4,000 proteins that are being treated by MSD. We have some samples that are really, really highly regenerative and some samples that are not regenerative at all from different bone marrow samples that we have. So we compared the regenerative bone marrows to the non-regenerative bone marrows, looking at all the proteins that they secrete and say, well, what's significantly increased in the regenerative ones compared to the non-regenerative ones. We also have the Wnt activation that we knew increases the regenerative capacity of the bone marrow derived mesenchymal stem cells. So we did another proteomic screen looking at Wnt stimulated cells to Wnt pathway inhibited cells. And what we did is we took these two screens, we found what was upregulated in the regenerative versus the non-regenerative. We found out what was upregulated in the Wnt versus the Wnt stimulated versus the Wnt inhibited. And what we found was that there were 9 candidates that were common in both screens that were highly upregulated in the regenerative and Wnt stimulated samples. And we're working with those, trying to work with those proteins using, exposing those proteins in the lab to human islets or injecting them into the pancreas of mice that have all of their beta cells knocked out to see which are the most important out of these nine candidates.
Okay. So currently, is there any theory favoring whether it's the alpha cells that can differentiate back into beta cells, or is it that progenitor cell idea with the progenitor cell in the ducts that's more favored currently?
Okay, well, I think at least in the mouse, a lot of these studies are done in mouse. I think the alpha to beta cell transition is quite well documented and quite well established now. And it's all happened over the past 5 years or so. So I think the alpha to beta cell transition is an important regenerative mechanism for diabetes patients. We think that this process is trying to go on all the time in patients, let's say with type 1 diabetes, but as soon as they regenerate new beta cells, they're destroyed by the autoimmune reaction that's characteristic of type 1 diabetes, what caused their diabetes in the first place. So the alpha to beta cell transition, but that process becomes exhausted. Again, I always use this term, regenerative cell exhaustion. So we need to find a way to dampen the autoimmunity and at the same time use the proteins or the MSC conditioned media to raise the level of regeneration. I think of it as a balance. So if we can raise regeneration and dampen autoimmunity at the same time, we might have the ability to generate new islets that are functional in patients with type 1 diabetes.
Okay. Okay.
We're not there yet, though. We're still working in mice. We're still trying to figure out the mechanisms. So the alpha to beta cell transition, I think, is very well documented in mice.
Okay.
And there are papers out there that says that this does occur in humans as well, especially in patients with type 1 diabetes.
Mm-hmm.
Now, what replaces the alpha cells is less well known.
Mm-hmm.
So we're sort of going out on a limb a little bit to say that perhaps it's the a facultative endocrine precursor cell that's associated with the ductal epithelial cells that can produce these new this pathway to produce new beta cells.
For sure. Okay.
But we have a long way to go to prove that.
Okay.
Years of research to be able to show that in mice first, and then to be able to apply it to humans. But we hope that someday we'll be able to get there, and there will be new therapies for type 1 diabetes from this research later on.